With 40% of Canadians receiving a cancer diagnosis in their lifetime, ultimately resulting in death for 25% of the population, cancer is truly a disease that has affected nearly everyone in this room in one way or another. Now, cancer's deadliness arises from a process called metastasis, which is when cancer cells break away from a tumor and then travel throughout the body to establish secondary tumors in new locations. But these tumors can be fatal when they're established in vital organs, such as in the lungs, liver, or brain. My research is centered on finding new drug targets for these aggressive cancers. But before we can get into that, we have to ask, what makes cancer cells so good at traveling these long distances? The mechanisms of metastasis are varied, but one place we can direct our attention is an internal structure that resembles the skeleton of a cell. This skeleton is composed of a protein network, and it's constantly being reorganized to help cells move, making it a good starting point for our search. Interestingly, genetic analysis has shown that there's a protein present at elevated levels in several different kinds of aggressive cancers, and it can modify this skeleton to help control how it's organized, making it a potential drug target. So I've been putting this protein under the microscope, both figuratively and literally, to better understand how it works and how it affects cancer cells. One way to understand how this protein works involves finding a way to inhibit its activity, but there are currently no drugs that can do that. So we've been collaborating with TMU's own chemistry department, along with an industry partner, and using advanced algorithms that can analyze protein structure, a list of potential drug candidates was generated and then synthesized. I then took this list and narrowed it down to a handful of strong contenders by assessing how well it affected my protein's activity. So now with these inhibitors, the next steps are exciting. To start, I've been treating skin melanoma cells with my drugs to see if there's a cell-wide difference in the amount of activity my protein displays. And so far, the results are promising, with drug-treated cells showing a dramatic drop in protein activity levels. Next up, I'll be taking an aggressive form of breast cancer cells and treating them with my drugs to see if there's a difference in the way that these cancer cells move and travel. My future goals will also be to study how these drugs bind my protein, which will then reveal where its activity is being regulated. So while there's still clearly a lot of work left to be done, the hope is that through research like mine, we can identify new therapeutic targets and put a stop to cancer on the move.